Perfect. So hi, everyone. So it was a real pleasure to be here in the first Artemisia Connect meeting. So I hope you enjoy the presentation and the paper that I will explain today. But before that, I will explain a little bit about myself, okay? And as Arnaud said, I'm from Spain. So I have a background in biochemist, a PhD in plant molecular farming, and a postdoc as well. So I have been working not only in Spain, but also in other countries such as India, Italy, Greece, or Colombia. Okay. So during my postdoc is when I start working with Artemisia annua many years ago. At the first moment, so I was working, as my background is, plant molecular farming, I was, working how to increase, the artemisinin content in Artemisia annua, how to increase the biomass, how to increase other biomolecules. But a few years ago, we created a company, a startup company that is called TricoPharming. So where we are working also, we are trying to elucidate other beneficial aspects of Artemisia annua, in this case, in the animal health sector, okay? So we are focusing on the pet, so dogs and cats sector. And so we are doing also a lot of research, not only to improve the agronomical aspects of the plant, but also to elucidate the different mechanisms, how Artemisia annua and how artemisinin molecule is working towards different pathologies or different disorders in the animal health. So that is why the topic of the paper that we published at the end of 2025, September, October, if I remember well, is published in a very specific animal health paper journal, Veterinary Science, and the title of the paper is Standardized Artemisia annua exhibit a dual anti-leishmanial activity and immuno-modulatory potential in vitro. Just to mention, okay, today also in the meeting, there is Stefania Morua, that is the first author of the paper. So even she can join later for answering the questions, okay? But in our research and development team of TricoPharming, we are not alone in this paper. So we have Stefania and Laura Cuyas from TricoPharming, but also, as you can see here in the authors, we have other authors that they belong to different departments in Spain, such as the Institute of Tropical Diseases and Public Health in Canary Islands, also Biomedical Research Networking Center of Infectious Diseases in Madrid, or the National Center for Food Technology and Safety in the north of Spain, in Navarra, okay? So just, I would like to acknowledge to all the authors that make this paper possible. So we go to the next slide, okay? You know that Artemisia annua is very famous because of malaria. Everybody knows that in 2015, the professor Jiu got the Nobel Prize because, because of, the potential of artemisinin in treating malaria. Indeed, Dominique later will explain about a paper of Artemisia annua and also Artemisia afra, I guess, regarding the potential of malaria. But since 2015, so more than 10 years ago, there has been a lot of interest, a lot of international interest regarding the potential of Artemisia annua plant regarding other pathologies. And that is why the focus of this topic of the paper is not malaria, but is leishmaniasis. I don't know if you have heard about the leishmaniasis, but leishmaniasis is an endemic disease, as you can see here in the map, okay, in either Asia, Africa, America, and Southern Europe. It is caused by Leishmania parasites and transmitted through sandflies, okay? So it's not the same as malaria. It's also transmitted by, sandflies, but by a different species of sandfly, and it's a completely different intracellular parasite. So what we know that in humans, so there is almost 1 million of new cases occurring annually, okay? And there are two different kinds of leishmaniasis. So it's the same disease, but with two different levels. So you have the cutaneous leishmaniasis that you can see here, the different strains that they are creating this disease. So like Leishmaniasis panamensis, Leishmaniasis brasiliensis, okay? So the cutaneous leishmaniasis is affecting mainly the skin and other, physical, superficial physical issues. I will not show you the pictures, okay, because they are quite disgusting, and we are, in the period of, lunchtime, so it's better not to show them. But also we have a more dangerous level of the leishmaniasis disease, that is the visceral leishmaniasis. That is more affected by leishmaniasis strains, such as Leishmaniasis donovani or Leishmaniasis infectum. And it's not only affecting the skin, but also is affecting internally the human or the animals. Okay, so if we go to the next slide, I will introduce where we focus. Okay, why? Because, okay, in human, leishmaniasis, as you saw before in the map, is affecting a lot of population, but luckily is affecting, the morbidity is very high, but the mortality luckily is very low. So the animal that is more affected by leishmaniasis is not a cat, is not a mice, is not a rabbit. They are also affected by leishmaniasis, but the animal that is more affected with leishmaniasis is the dog. So the dog is a natural reservoir of the disease, and the main problem that, there is a high mortality of the disease of dogs affected by leishmaniasis. So if we focus on dogs, and this is the topic a little bit of the paper, so canine leishmaniasis is primarily caused by Leishmania infantum, so one of the strains that I mentioned before in the previous slide, is endemic in over 90 countries with an estimated 700 million infected dogs, so including 2.5 million cases in Europe, in the south part of Europe. So that is why I'm coming from Spain. So the southern part of Europe, all the Mediterranean area, is very well affected by Leishmania infantum in dogs. And that is why many, many years ago, we started this research project, okay, in order to elucidate if Artemisia annua is able to help the dog affected by Leishmania infantum, okay? So Leishmania replication is in the macrophages. Macrophages are the immune system cells. So in a simple way, macrophages, they eat the bad stuff, okay? So they are eating the bacteria, viruses, the dead cells, and also they are able to release cytokinins that they call other immune cells. So macrophages, they are one of the main immune system cells that, and the replication of this intracellular parasite is happening there, okay? So that is why canine leishmaniasis is a chronic disease. So once the dog is affected by leishmaniasis, will have it forever. So that is why it's very, very important also that, the immune response Th1, which is the cellular immune system, is very strong because, the Th1 response is the one that facilitates the macrophage activation and the parasite control. So we'll show the picture of the dog because they are not that bad as the human ones. So here you can see the main symptoms of the main injuries of a dog that is affected by Leishmaniasis infantum. Of course, you can see this kind of skin injuries. You can have also this kind of ocular injuries, as you can see there, that even the dog can become blind. And also a very typical symptom of leishmaniasis in dogs is that there is a really long growth of the nails, as you can see here, the picture below of the dog. So these are the first symptom that the dog is having dermatological issues regarding of the leishmaniasis infectum, okay? So this is why you are able to control the disease. So at the earliest phases of the disease, what's happened is the leishmaniasis is becoming visceral, so it's affecting the kidney, it's affecting the liver, it's affecting two other organs, is when the mortality of the mortality is really increasing. So it's very, very important to control the disease in order not to become visceral and that the dog will die because of a, kidney or a liver failure, okay? So keeping that in mind, as I told you, from 2015, a lot of publications they are revealing the high potential of Artemisia annua, not only to treat malaria, but also to other diseases such as cancer. There are plenty of publications regarding cancer. So I hope in the next meetings, Artemisia Connect, some people will explain some papers regarding cancer. But also there was some papers regarding the potential of Artemisia annua and artemisinin towards different strains of leishmaniasis. Everybody knows that, as you can see here in the figure, artemisinin contain a natural endoperoxide bridge that, upon activation or cleavage, generate cytotoxic reactive oxygen species to which leishmania parasite, due to its limited antioxidant defenses, is particularly vulnerable. Okay, so how we know? The little information we know is that artemisinin, when breaks the natural endoperoxide bridge, will generate a lot of reactive oxygen species that will kill the parasite. Okay, but everybody that is here, think that Artemisia annua is not only artemisinin, and also I'm very confident of that. Okay, there are other molecules. Artemisinin is a sesquiterpene, but there are other terpenes, there are other flavonoids, other polysaccharides, and other coumarins that they play a pharmacodynamical synergy with artemisinin. Okay, in other words, so for instance, there are some flavonoids that they are helping artemisinin to be more bioeffective, or to increase also the life cycle of the molecule artemisinin. Okay, so this is very important, that Artemisia annua, as we say, is not a monotherapy, it's a polytherapy. So there are other molecules involved in improving the health state of the dog or of the humans, and also there is other playing other roles, other biochemical roles in the host. Okay, but we want to focus in artemisinin in this paper, and you will understand why. Okay, why? Because artemisinin has shown already promising efficacy against different leishmania species, such as leishmaniosis, Leishmania donovani or major. But this paper is the first time that Artemisia annua is tested against Leishmania infantum. So also important to keep in mind that not only there is some publication regarding the potential of Artemisia annua to treat leishmaniosis, but also to modulate both the innate and the adaptive immunity. Okay, so Artemisia annua and artemisinin specifically show the ability to modulate both immune systems by enhancing the macrophage function, reducing the oxidative stress, and regulating the T cell responses. Okay, so we want to measure also if Artemisia annua how is going to affect the Th1, the cellular immune response in the body. Okay, this to keep in mind, that this is important to keep that the pro-inflammatory cytokinins, they help to coordinate the defense against different infections and injuries. So this is the case, as you can see here in the figure, of interleukin-6 and TNF-alpha. Okay, but what's happened? If the levels of the pro-inflammatory cytokinins, they are very high, in the case of IL-6, they are associated and increased disease severity in chronic diseases, okay, such as chronic leishmaniosis. Why? Because if you have very high levels of interleukin-6 all the time, okay, it will suppress interferon gamma production, will enhance B cell activation, contributing to a systemic damage. So it's very important to keep control the pro-inflammatory cytokinins, not only interleukin-6, but also TNF-alpha. Okay, so keeping in mind all these two informations that we have previously before publishing our paper, our research started like that. Okay, we wanted to evaluate it. We wanted to answer three different questions. First, there is a possible double action, so immune modulator and anti-leishmanial effect of Artemisia annua, yes or not? This effect is artemisinin independent, or there are other molecules within the plant that play also a very important role? And the last questions, and there is an efficiency in Leishmania's infantum. Okay, so there was the main 3 questions that we wanted to answer during, at the beginning of the research, and that we answer here in the paper, and I will explain you later, okay? So we evaluated the in vitro anti-leishmanial and immunomodulatory effects of Artemisia annua extracts against the different Leishmania infantum strains, okay? So just to mention that all the material was obtained and was produced in our farm that we have in Tenerife, in Canary Islands, in Spain, okay? We use different material of Artemisia annua with different concentration of artemisinin. Why? Because we wanted to elucidate it if artemisinin is the main molecule to play this effect, okay? So we created different extracts with different concentration of artemisinin, ranging from 1.21% to 0.13%, okay? And we test all these extracts in promastigote and amastigotes of Leishmania infantum, and also in macrophages in order to measure the immune response, okay? And it's something that I will explain you later in these slides, okay? Okay, I don't know what's happened with the slides, that they're quite, I download and there is some small mistakes, sorry for that, okay? But okay, first, we measure the anti-leishmanial effects of Artemisia annua in Leishmania infantum promastigote. Promastigote are the juvenile phases of the Leishmania intracellular parasite, okay? So what we measure for that? We measure the IC50, that IC50 stands for half maximal inhibitory concentration. So IC50 is a quantitative measurement used in pharmacology, biochemistry, and toxicology in order to indicate how much of a substance is needed to inhibit a biological process by the 50%, okay? So in other words, the lower the IC value it is, means more potent is the compound, okay? So keep in mind that the lower is the IC50 value, more potent is the compound. So here we measure the IC50 of 3 different extracts of Artemisia annua. As I told you, 21%, 0.75%, and 0.13%. And here down, you can see values, okay? So you can see that when the Artemisia annua extract has a low quantity of artemisinin, 0.13, so the IC value, it was very, very high. So even 800 micrograms per milliliter, which is the meaning of that, okay? That Artemisia annua extracts with low concentration of artemisinin, they have a very little effect in killing the Leishmania infantum promastigote. So this effect was higher when higher was the concentration of artemisinin. So for instance, the concentration of artemisinin was 0.75%. The effect of the extract was 4 times stronger. In the case of 121, it's from 231 to 58 micrograms per ml. Which is the meaning of that? Okay, more artemisinin levels has the plant, higher is the leishmanicidal activity of the extract, okay? So keep in mind that. And the effects, they were very, very, very clear. Similar results, we got it not only in the Leishmania infantum promastigote, but also in the amastigote, okay? Amastigote is the adult phase of the parasite. That is the one that is affecting the macrophages, as you can see here in the picture, okay? So here we measure also, we selected the extract that has a higher concentration of artemisinin, 121, and what we saw that the IC50 value was also very low. So meaning that the 121 artemisinin extract exhibits an inhibitory activity against the intracellular amastigote states. And here you can see here in the picture, one of the top, the control. The spots that you can see here are all the parasites that they release from the infected macrophages. The one below is after the treatment of Artemisia annua, where you can see that there was, the parasite load has decreased a lot. Okay, so this is very, very important information. But as I told you before, it's not only important to control the disease, but like a dog and a cat, when they got the disease, it's a chronic disease, you have to keep always in a good way, in terms of effectiveness, the immune system. So why? And specifically the cellular response. Why? Because it's the one that predisposes the body towards viral, fungal, and parasitic infections. So if you keep strong the Th1 cellular immune system, your body will be stronger in order to fight against the parasite of leishmaniasis, and also the parasite of malaria, and also other viral and fungal diseases. Okay, so this is important to keep in mind. Why? Because a way of controlling and make it stronger the cellular response is to control the pro-inflammatory cytokinins. Okay, why? Because these pro-inflammatory cytokinins coordinate the defense against different infections and injuries, but when they are produced in excess, so when there is an infection going on, malaria, leishmaniasis, and other kind of infections, so the uncontrolled synthesis of these pro-inflammatory cytokinins could have negative effects. So immune system imbalance, chronic inflammation, autoimmune disease, and tissue damage. So that is why I will go faster in this way in order not to make the presentation longer. We also measure the concentration of pro-inflammatory cytokinins, interleukin-6 and TNF-alpha, at different concentration of artemisinin. Okay, this we did it in macrophages in vitro, okay, under inflammation conditions using LPS, which is the liposaccharide, okay, that is creating inflammation, okay. As a positive control, we use hydrocortisone. That is working very well, but also has a lot of negative side effects. And as you can see here, when you use 0.6 percentage of artemisinin, the Artemisia extract, we're able to decrease 45% the interleukin-6. Why? If the concentration of artemisinin was higher, the effect was much better. As you can see here, we're achieving the 55% of reduction in the interleukin-6 levels. Something similar happened in the case of of the other pro-inflammatory cytokinin, TNF-alpha, okay, but the results, they were even more clear. In the case of the extract of Artemisia annua with 0.6 of artemisinin, the reduction of TNF-alpha levels were only 25%, while in the case of 0.9% of artemisinin, the reduction, as you can see here in the picture, was above 80%. Okay, so in conclusion, and just to finish, okay, there was a very clear correlation between artemisinin content and anti-promastigote efficacy. Also, there was a very strong anti-amastigote activity on the extract of Artemisia annua that has a higher concentration of artemisinin, and also Artemisia annua reduced the pro-inflammatory cytokinins, enhancing the immune efficiency while minimizing inflammatory tissue damage. And also, in this case, it was a very clear artemisinin dose-dependent relationship. Okay, so this is the conclusion of the paper. I make it probably a bit longer than I expected, but okay, I hope it was interesting, it was clear for you. Anyway, if not, I will be here answering for any questions you want. And thank you very much.
Thank you. Thank you very much, Luis. I'm adding Dominique, who will ask you the first question.
Or I have to write?
Yes. We can hear you, Dominique.
I can speak?
Yes.
Okay. So, well, thank you very much, Luis. And I was, of course, very interested in the immunomodulation by Artemisia, the Th1 response, and so on. You said that artemisinin is what is important in Artemisia.
I mean, it's not the only important, but here we measure that it was.
Yeah. But wait. But you said also that there is other things in Artemisia, which maybe can be efficient. And so, did you try, for example, Artemisia afra, what we did in malaria, where there is very low quantities of artemisinin? Did you try that?
No, to be honest, no, because we focus in Artemisia annua. Maybe in the future we can do that. I wish if we get the budget, I will be more than happy to measure about Artemisia afra. And I agree with you. So there is even, in the phytochemical profile of Artemisia annua, there are other molecules such as, flavonoids, eupatorin, castesin. They play a very important role in the immune system. Also polysaccharides, coumarins. So here in this paper, we only measure artemisinin and some flavonoids in order to see that it was the same concentration. So it was very clear that it was a very, correlation with artemisinin, but we didn't measure all the molecules. So maybe there was another molecules that they were also increased and that they are important also for the immune system. Okay.
Okay, well, thank you. So I have just to begin the conversation. Maybe there is other question from
Yes, I don't see questions right now on the comments, but please. Oh, that's a question of Langong Methodius. Can you see it, Luis?
Sorry?
So Langong Methodius is asking a question.
Methodius. Hi, Methodius. How are you doing?
Saying thank you for the nice presentation. I am Methodius from the University of Liège, Belgium. I wish to find out if you did compare the activity of the Artemisia annua extract with that of the standard drug.
Well, we didn't do it for this paper, but I have to say that now we are running another in vitro experiments comparing with artemisinin and other pathologies, and the effect seems to be higher in the plant. So meaning that there are other compounds important for that. But in this paper, we didn't measure that.
Good. Also, what kind of values did you use to compare the activity level?
What do you mean by cutoff values? I mean, statistical things, or it's important. It's difficult because I have to write the question. Maybe we can keep later. So when, after the presentation, he can write all the questions and I can answer later, maybe.
Yes. Diana Mateud is asking, very nice data, thanks. Have you tried it in vivo?
Indeed, right now we are doing 2 in vivo trials with dogs affected by leishmaniasis. We started the last year. Yeah, so hopefully in the near future we'll be able to present all the in vivo results we have regarding Artemisia annua and dogs affected by leishmaniasis.